Mammalian longevity is a direct function of tissue rejuvenation. This repair process is driven by specific populations of adult stem cells, or ASCs, which are responsible for maintaining organ and tissue integrity throughout a lifespan. The total count of these stem cells is essentially fixed at birth. They are geographically anchored in specific niches. Over time, factors like UV exposure, pollutants, and chronic infections progressively deplete these localized reservoirs. As the ASC count drops, the body's capacity for perfect repair fails. This structural deficit is what manifests macroscopically as tissue degeneration, organ dysfunction, and the formation of permanent scars. In this model, biological aging is the cumulative depletion and eventual failure of the localized adult stem cell reservoir. In 2006, the development of induced pluripotent stem cells, iPSCs, offered a potential way to artificially replenish these stem cell pools using 4 genes known as the OSKM factors. However, the OSKM method intrinsically couples cellular reprogramming with a hyper-accelerated cell cycle, forcing division every 16 to 18 hours. This rapid proliferation carries an inherent risk of triggering teratomas, disorganized tumorous tissue masses, when introduced into a living organism. Additionally, OSKM induction resets cells to an embryonic state, rather than generating the specific adult stem cells required for targeted, localized tissue repair. For cellular reprogramming to be clinically viable in vivo, the process must decouple pluripotency from hyperproliferation and bypass the OSKM pathway. Even when 2020 refinements reduced the sequence to 3 factors to mitigate tumor risks, the mechanism still could not produce targeted, multipotent adult cells. However, a distinct alternative emerged from Dr. Shilung Lin's laboratory in 2008. Instead of utilizing the Yamanaka proteins, his team deployed an embryonic-specific microRNA called miR-302, bypassing the OSKM factors entirely. In mammalian biology, the maintenance of stem cell pluripotency is naturally handled during the maternal zygotic transition, the period immediately following fertilization. During this early phase, maternal RNA molecules deposited in the oocyte act as inhibitors. They prevent premature cell differentiation by simultaneously regulating over 445 specific cellular genes through RNA interference.
But very quickly, those maternal RNAs, which are mostly from the MIR290 family, start to degrade. So to stop the embryo from prematurely differentiating into specialized cells, the zygote has to start producing its own regulators. And in humans, the MIR302 family quickly tags in to replace those maternal RNAs. It steps up and flawlessly maintains the stem cell pluripotent state all the way up to the 64-cell stage. And the best part? It does all of this without causing even a single trace of cancer.
This sequence tracks morphological progression from the 1-cell stage to a mature embryoid body. By exposing adult somatic cells to miR302, researchers replicated the maternal zygotic transition. Histological cross-sections confirm their ability to differentiate into tissues across all 3 primary germlines: ectoderm, mesoderm, and endoderm. To safely repair aging tissue, the scientific focus shifted from the volatile state of artificial pluripotency to the targeted, stable induction of adult multipotent cells. This diagram illustrates exactly how miR302 achieves safe cellular reprogramming. It operates through a simultaneous dual-action mechanism. On the left, the reprogramming cycle activates. The microRNA silences specific epigenetic regulators, triggering wide-range global DNA demethylation across the cell. This demethylation effectively wipes the epigenetic slate preen. It resets the gene expression pattern to a plastic, younger state. Simultaneously, the tumor suppression cycle engages on the right. Here, miR302 silences the CDK2 and cyclin D cell proliferation pathways. By suppressing these oncogenes, the cell cycle is forcibly decelerated back to a safe rhythm of 22 to 24 hours. This built-in break actively prevents cancerous transformation. By decoupling the cellular rejuvenation process from accelerated growth, this dual pathway provides a tumor-free environment required to safely induce CD34-positive adult stem cells. To test this in a living system, researchers utilized a mini-pig wound healing model, selected specifically because its epidermal architecture closely mimics human skin. This graph maps wound closure rates. By day 11, tissues treated with formulated miR302 or iPSC lysate reached a 90% closure rate, outpacing controls. Crucially, in the first 3 days, treated wounds exhibited over 20% contraction, the prerequisite for scarless tissue regeneration. This early contraction phase is a biological requirement for perfect healing, as it prevents disfigurement and the formation of scar tissue. Natural adult stem cells follow asymmetric division, generating limited replacements. But MIR302 stimulates a novel symmetric division, rapidly amplifying the stem cell population. We see the physical proof of this symmetric expansion in this immunohistochemical stain. The bright green clusters are induced CD34-positive adult stem cell pouches, spreading across the healed tissue, forming localized colonies roughly 240 to 290 micrometers apart. MIR302 triggers targeted amplification of local adult stem cell pools, providing the cellular material required for complete structural regeneration. Under normal conditions, natural CD34-positive cells follow asymmetric division rules, meaning one stem cell divides into one replica and one specialized tissue cell. This inherently caps their ability to repair large-scale trauma. The MIR302 formulation overrides this natural limit. It forces the stem cells into a sequence of symmetric division, where one stem cell divides into two identical stem cells, resulting in rapid exponential amplification. Forcing adult stem cells into this state of geometric expansion provides the exact mechanical engine needed to fully regenerate tissue without leaving a scar. Cultivated in vitro, these isolated CD34-positive colonies actively expand. They uniquely generate a thin, isolating membrane layer, dramatically streamlining purification. Once purified, researchers selectively removed specific growth factors, reliably directing these multipotent stem cells to differentiate into specialized neural and mesenchymal lineages. To prove viability in a living organism, the amplified CD34-positive cells were tagged with a red fluorescent protein and implanted directly into immunodeficient mice. After a few weeks of incubation, the red fluorescent markers confirmed the derived cells successfully integrated into brain and bone marrow tissues. This viability extends across diverse systems, as injected cells also formed distinct functioning pouches within cardiac muscle, skeletal muscle, and gut epithelium. This expansion provides a source of highly viable, multipotent raw materials capable of structurally repairing 5 distinct mammalian organ lineages. Before any cellular reprogramming factor can be considered viable for clinical use, it must demonstrate absolute systemic safety and precise biodistribution within a living body. These 4 cross-sections display the results of intravenous injection into the circulatory system. The red TAMRA labeling indicates that the formulated MIR302 distributed effectively through the heart, blood vessels, spleen, and bone marrow, with 0 evidence of triggered tumorigenesis. To conduct an absolute stress test regarding cancer risk, researchers injected the MIR302 formulation directly into a highly malignant human hepatocarcinoma tumor xenografted into a mouse model. These macroscopic photographs capture the physical results after just 4 weeks. The large, dark tumors from the placebo group stand in stark contrast to the treated tumors, which exhibited a massive 90% reduction in average volume. This mechanism represents cancer reversion therapy, a process distinct from the cell-killing mechanics of traditional oncology. The microRNA directly reprograms the epigenetic state of the high-grade malignancy, forcing it to revert back into a structurally benign state. The microscopic architecture proves the transformation. We can identify newly formed central vein and portal triad-like structures, mirroring healthy liver lobules. By activating its dual pathway, MIR302 actively suppresses oncogenesis. It takes the largest historical risk of stem cell therapy, cancer, and turns the mechanism itself into a targeted therapeutic tool. To measure the ultimate physiological impact of this mechanism, researchers compiled data from a comprehensive 14-year longitudinal study, focusing on the systemic anti-aging effects of MIR302 in mammals. This Kaplan-Meier survival curve tracks the primary finding. Mice treated with systemic MIR302 reached an average lifespan of 64 months. This is more than double the 28.8-month average of the untreated control group. The control mice died of typical systemic failures, primarily cardiac hypertrophy and bone marrow failure. By comparison, the tissues of the MIR302 subjects remained functionally preserved at 74 months, with no detectable lesions in the heart or bone marrow. The evidence suggests this structural preservation is the direct clinical result of symmetric stem cell expansion. By continuously replenishing the adult stem cell pool, the body is given the biological resources to outpace standard age-related cellular attrition. The data mandates a pivot in clinical strategy. The scientific horizon is no longer tied to the inherent risks of artificial pluripotency, but rests firmly on the safe, systemic renewal of endogenous adult stem cells. This specific expansion mechanism enables immediate therapeutic applications, treating intractable wounds, managing aggressive malignancies, and reversing severe degenerative organ illness. By utilizing MIR-302 to induce symmetric expansion of our biological repair systems, we possess a viable molecular framework to extend human health span.